Yeah, so good evening everyone. So today we are in the week 9 of the live session of which an Interactomics course of which mostly we will be talking about the level 3 techniques. So here mostly the major thing that have been focused on the two technologies, that is basic layer interferometry or the system in which the white light gets changed with the interface and the interaction. So study the interactions, how strong between two molecules are, two biomolecules are. Whereas in the another important is to have coupling of different technologies together for better understanding of interactions. For example, the SPR imaging. So we would be looking at that as well in a great detail. So one of the applications of the level 3 technologies that has been spoken about or that have been uh, talked about is like the high spatial resolution, which measures the simultaneously over the entire area radiated by light detector array and CCD, and potential to combine with micro. So we know that what happens in SPR. So SPR generally in that we have that ligands which gets immobilized and it's being measured of the response units or changes in the resonance response units. But here if we have whole microarray like stuff, here we will have one source of light and which will be coupled with a CCD camera. So this will be get detected and will capture that whole. So this is one which can be used as a whole for which is the SPR imaging, of which we can look at certain SPR imaging principles. So in an SPRI system, a coherent polarized light beam so a polarized beam light is used instead of the polychromatic light. So first thing that is different is that a light source where it is being used a coherent polarized light but not a polychromatic light. This change helps expand the light cover on a larger area of the sensing surface, and the reflected light is captured by a charged coupled device CCD camera for further imaging analysis. So which is used for a further imaging analysis, right? So this first difference is that coherent polarized light and then reflected light by a CCD camera for further imaging provides images in real time, hundreds of active spots. Measurement is stringently constant wavelength and a constant angle. Okay, so at a constant, so here we can see that. So flow cells include layer, prism, in this specific interaction with the target molecules. There's a fixed incident light, right? So So this is just the improved version of the SPR machine, right? So what thing we can do is SPR imaging principles. Yeah. So this was one which I wanted to give you. So it's an level-free optical detection technique that is used to monitor and analyze biomolecular interactions in your real time. Enables users to visualize the entire working area and work in a multiplex format, not in a single place format, but rather in a multiplex format. So what does multiplex mean? Means that the different types of ligands can be mobilized on a single SPRI biochip. So multiplexing means that different types of ligands can be mobilized on your single SPRI biochip. And it also enables the study of many parameters at the same time: concentration, immobilization, pH, etc., to compare, rank, and select molecules easily. So different types of ligands can be immobilized and enables the study at the same time that concentration, pH, etc. So SPRI, what is information is full characterizations, which in turn sample solution. So SPRI is used to monitor the refractive index on the basis of the SPRI biochip. Binding event, mass accumulation induce a change of refractive index and a shift of the position of the resonance angle. SPRI follows the variation of reflectivity occurring at a fixed angle versus their time. Baseline association, equilibrium, dissociation. Immobilized when the sample solution is injected. So of whatever video we had just seen, it is exactly the same procedure what it is mentioned here. So molecular binding can occur, shift in the plasmon curves, and then increase in the reflectivity. I show the variation of reflectivity versus time association phase. Also be monitored on the SPRI difference image. When the sample solution leaves the flow cell, the ligand analyte complexes dissociate. This indices a shift of the plasmon curves and a decrease of your reflectivity. The kinetic curve shows the variations of reflectivity versus time. The process can be monitored on the SPRI difference image as the interacting spots become darker. And finally, when all the ligand and analyte complexes are fully dissociated, and if you just the initial set, the difference image is black again, right? So one of the example here, gold film polarizer light. So polarized light is used other than the polychromatic light, and in the gold film there is that human vesicles which is reflected and is getting into the camera, and you can see with different instances it can be used. So that you all know the SPRs, but what we are so surface plasmon resonance imaging, robust technique for visualizing the refractive index changes. So I'm trying to give you whatever principles that you can understand it in a much better way, which enables researchers to observe interactions between nanoscale objects in an imaging manner. In the past, scholars have been attracted by the prism coupled and non-prism coupled configurations of SPRI and published numerous experimental results. Principle of SPRI and discusses recent developments in prism coupled and non-prism coupled SPRI techniques in detail. The purpose is to briefly summarize the recent advances, provides an outlook. So this is how virus, bacteria, cell, exosomes, biomolecules, biological applications, prism, light, and we can get it. So is an important technology between the nanoscale targets. With the advantages of high sensitivity, label-free sample, simple operation, and the rapid detection. SPR standouts from many biological detection technologies, which has aroused the strong interest of researchers. Traditional SPR systems are mainly based on spectral analysis, basic way of detecting SPR signals. Through the charge of the signal, the interaction between the sample and the modulation that can be quantified. Become a robust technique for visual analysis. Then we have into two types. One is prism coupled and non-prism coupled. So it can be prism coupled. In prism coupled, SPRI can be further split into the traditional prism and the non-traditional prism. Intensity, wavelength, angle, phase sense, and all those. Major impact. So principle of this is what we mostly we would be looking for. So basically, the researchers took different configurations. For the slit and the metal, the width of the slit. For the placement structure, the metal field is calculated. Incident light and the reflected light. So don't go into this particle scattering method. So this is more about this thing, angles. Yeah. So the recent advances in the surface plasma resonance imaging sensors. So you can just look at it here. So it is a charge density oscillation that may exist at the interface of two media or with the opposite dielectric constants of the opposite signs. Surface plasma can consist of a semi-infinite metal with a permittivity and with this thing transverse polarized wave. Surface is an emission wave. So these are the configurations. So this is regarding mostly the sensors and the instrumentation part, of which if you are interested, you can look at about. So the same thing. So this is what basically more or less about the SPR and the SPR imaging systems with some things you have to understand the instrumentation part. If you are interested, you can go through it, but mostly the principle is the same. So the shape which we gives the kinetics, concentration, amplitude will give the surface capacity molecular weight. So SPRI's data processing includes a double reference subject response from the reference surface, subtract response of the buffer injection, and the data analysis includes global fit data sets in the appropriate fitting models. So what is the advantage of SPRI or uh, surface plasma resonance imaging? You can talk about is that less reagent consumption, same solution. That is your clinical sample drugs and used to monitor thousands of spots simultaneously, time saving and the cost saving, and buffer temperature with each improvement in the measurement reliability. Yeah, so now we would be transitioning into the mass spectrometry coupled interactomics. So if you have any question till now, and we'll start with this in a few minutes. Yeah, so any question? No, right? Okay. So we will be now looking at an IPMS of or a case study for immunoprecipitation coupled mass spectrometry for static interactions. So it has been a Alzheimer's IPMS. So just give me a meeting. One second. Hmm. So this is the paper that we were looking at. Hopefully, invisible. Yeah. So, so this is the thing, multi-omics profiling of Alzheimer's disease serum for the identification of antibody biomarkers. So we have spoken a lot about uh, one second. So this is multi-omics profiling of Alzheimer's disease for that antibody biomarker. So we have spoken a lot about in the initial weeks when we had started discussing about the interactomics. So is that uh, So for the identification of the autoantibody uh, biomarker, so that when the disease persists, there is a change in the proteins and the protein expressions, so which leads to the body recognizing it as a foreign proteins and all and generating the antibodies. So here, like the new biomarkers of Alzheimer's disease in preclinical are identified. Serum autoantibodies are potential candidate biomarkers. There we have identified the serum using protein microarrays and mass spectrometry based methods. Untargeted high density and low density planar protein epitope signature arrays and immunoprecipitation. So this is a classic example of what interactive tools and studies can do to help you in understanding and how many things you can get to be validated. Hello based amino acid tag. I will take this thing attenuated or antigens identified by a multi-omics approach, new insights of the disease, and should help in your blood-based diagnosis of your Alzheimer's disease. And so this is the data availability. So the main cause of dementia, 30%, chronic and remitting neurovirus, accumulation of intracellular tau. So this is for the AD diagnosis. What we will be going into the mostly for the IPMS, right? That IP coupled to LC-MS if you can see here. So this one is our major interest. So let's see how it goes. So we will be looking majorly focusing on this part, which is related to the brain extract preparation for IP coupled to LC-MS and WB, so or Western blot. So for the brain protein extracts used in immunoprecipitation coupled to liquid chromatography and tandem mass spectrometry, prefrontal cortex tissue samples were cut on dry ice. So basically, the protein is being extracted. So these are the your what I say that uh, uh, protein is being used for finding the autoantibodies. So we were using the normal whichever normal proteins are there. Supplemented mechanical disaggregation. Quality was after 10% is test-based protein concentration determined by BC or TRP quantification method, anti-alpha tubulin antibody. Right. So now immunoprecipitation of protein targets of AD autoantibodies. Protein extraction from 8 prefrontal cortex tissue samples of AD patients was performed, and total of 550 microgram of each extract were pooled in a final volume of 800 microliter. A pool sample was then pre-cleaned for 1 hour at RT on rotation using a protein G spin tube column previously that have been equilibrated with the lysis buffer, and for equilibration lysis buffer were added. The buffer was removed. This process was repeated. The pre-cleaned protein sample was recovered and saved until use. IgGs were isolated from the serum and covalently coupled to protein G, and two sera pools, one from AD patients and another from healthy subjects, were prepared. Then two protein G steel traps were equilibrated. After equilibration of each tenfold diluted in TBS, each column, and incubated for 1 hour at rotation. After centrifugation, so I'll tell the concept after I'll go through the protocol. Columns are washed 10 times with wash buffer. For each wash, 400 microliter wash buffer were added, mixed up and down with the protein G resin and then spun. Okay, and with column and incubated at 1 hour at rotation and RT on rotation. After centrifugation, the volumes were washed 10 times with wash buffer. For each wash, 400 microliter wash buffer were added, mixed up and down with the protein G resin and spun. The wash buffer was changed. I will tell you the concept what is happening if you are not able to follow. So, and the wash buffer was changed after spinning. 400 microliter DMP was added for efficient antibody protein G cross linking. The protons were washed and later blocked, and then finally the subjects along with cool den extracts of 400 micro were incubated overnight after spinning. Uh, Finally, the protons were washed in test with water bound properties 3 times and then uh, was neutralized ABC for subsequent delivery. All the centrifugation steps were performed, saved and analyzed by STS page and commercial or silver staining. So before going into that, so what they are trying to say, just review. So first thing they had taken 80 patients, right? And you are extracting the proteins out. Right? Why this has been done? Okay, so 80 patients and then extracting the patient proteins out. So why? Why it is so? Because these, if any protein has developed any kind of antibody, so this is those proteins. Among those proteins only, the antibodies will be generated, right? Now, what in another scenario, they have taken right. So what it is there? So you have taken the beads. So these are protein G, protein A or G agarose beads, right? Right, protein A or G agarose beads. So these have affinity towards IgG. Now all the IgGs from serum IgGs are pulled down using these beads, right? So all the IgGs will be. Now what they say? So what it is happening? So once the beads are being pulled, so once these beads are getting pulled down, these IgGs. Now what will happen is that we will incubate these proteins with IgGs. Now, if just think of the concept and try to imagine if your IgGs or the antibodies is of any foreign protein or foreign particle that has been generated, so it will not be a part of this protein pool, right? Not this protein pool, bacteria against bacteria protein or viral protein or any kind of other infectious particles proteins, but for this protein pool, any IgGs if it is there, then that antibody and protein will get interacted. So this incubation is done with the beads which has pulled the serum IgGs and with these particular proteins. So if there is any autoantigen of whose autoantibodies have been generated, so if you have an autoantigen for which any kind of autoantibody has been generated, then what it will do is that it will go and bind with the IgGs, which you need to make sure that there is no non-specific interaction or non-specific binding. Rather, a strong binding happens, which you will be wash it out and elute it. So the antibodies, autoantigens, and their interactors will make a chain, and that you will be running in the gels, and you can uh, do the LCMS analysis. Right? So this you will be extracting the protein out, and then you are getting the autoantibody markers, and then finally you will be visualizing it in the gel, that interactors, and then you will be getting the proteins out. So now we will see that how the LCMS analysis has been performed. Right? So again, just now think, so I have given you the concept of basically that extracting out the protein pool because of interacting with the antibody. So if there is any protein, for instance, from outside, which is not of this particular antigens or antibodies, sorry, then definitely that happens is that it will not interact with the protein pool that have been there. Okay, so the protein pools that we have got is will not be there. So what we get it out is that the proteins coming out, that will be a different sets of proteins or antibodies. But when we have those particular proteins out, so among these proteins, if there is an autoantibody there of an autoantigen, it will interact and it will give us the result, right? So these interactors and those proteins are what we will be surely getting off. Now, so this is the reason why protein extraction has been done, and it has been done, and ethanol, triethanol for blocking, and DMP for stronger interaction and washing, and all these are different chemicals are being used to make these interactions and get it in the gel, which can be used for a LC-MS analysis and the data processing. So half of the eluted fraction 1 and stained, cut into 3 slices and separated unstained with 50 millimolar ABC, 50% ACN, dehydrated with ACN, and dried, reduced to DTT, a simple concept of bottom of proteomics for uh, basically breaking the proteins to peptides. And then gel pieces were dried and incubated, peptides were extracted, purified using a zip tip, and dried. Finally, samples were reconstituted and 1% acetic acid, 2% ACN before the analysis by in the proteomics for each analysis into a pre-column and diluted C18. The mobile phase was different solvents and the gradients, and this was done finally. Right? So now results. So you can see right? So now IPMS. Autoantibodies evaluated remarkably. So let us check once again from the first thing. Using planar and IPMSMS is different board with screen large planar arrays, education, breast. So these are the proteins that have been high confidence proteins that have been identified after IP coupled to SLC-MSMS for autoantibody profiling. So these have been identified, right? And then these autoantibodies validation and all those things. Basically, this is how you are analyzing and getting it. Okay. Yeah. So if you have any doubt, please let me know. The session will continue till 8 p.m. Uh, yeah. Thank you. Okay, so thank you for joining. We will meet on next week.